Hi, everybody. Welcome to another Chats with Chaudhry. It's a bank holiday Monday here in the UK, and it's wet and cold outside my window as I look right now. But hopefully, my day is going to be brightened up because today I'm joined by Brad Carlin, who's the Senior Advisor, Data Science and Statistics at PharmaLex. And we are going to be talking about, and bear, this is quite a long winded title, so you'll have to bear with me, the use of Bayesian and other novel statistical methods in clinical research and regulatory science, with emphasis on methods for rare and pediatric disease. So hopefully I've covered that all, Brad. So how are you, my friend? And hopefully it's sunny where you are, because it's horrible outside my window right now.
I'm great. It was a nice sunny weekend here in the Midwest, part of the United States, and it's great to be with you today.
Cool. And whereabouts are you calling in from? You said the Midwest, but whereabouts are you actually calling in?
I'm actually based in Minneapolis, Minnesota. So spring has finally come here. Things are looking up, and it was a lovely weekend here in Minnesota.
I'm glad to hear it, Brad. Well, at least you brought a bit of sunshine into my life, because I tell you what, it's cold right now.
Statistics always does that. Everyone feels happier when they think about statistics for a while.
Well, actually, it's a subject close to my heart, because people may not know this, but I did my degree in economic
Ah,
statistics, even if I can't say it properly any longer. But so I actually do enjoy the topic. So I look
good.
forward to talking about that. But before we start talking about statistics and Bayesian statistics, perhaps you can tell people a little bit more about PharmaLex and a quick overview on your own background as well.
Well, let's see. PharmaLex is a large multinational consulting firm that handles pharmaceutical consulting from really soup to nuts. It starts out doing, we do preclinical work, we do phase 1, phase 2, phase 3 clinical stuff. We do post-release studies. We do a whole lot with CMC, chemical manufacturing control, shelf life studies. And we do use a lot of Bayesian statistical techniques, which are fairly novel, fairly unusual amongst our competitors, but becoming more and more common.
Cool.
My own background was I was trained as a Bayesian statistician way back in the 1980s sometime at the University of Connecticut. And I took a job at the University of Minnesota and was an academic for most of my career, including chairing the department for 7 years, Division of Biostat at the University of Minnesota. And then went out on my own and worked 2 years on my own and joined the PharmaLex team about 6 months ago. And it's been a lot of fun so far.
All right, great. Fantastic. Well, congratulations. And I hope the new role continues to grow and be successful for you. So
Loving it so far.
fantastic. So let's talk about Bayesian statistics. A really basic question. What are they? And why do biopharm statisticians need to know about them?
So Bayesian statistics is actually a lot older than traditional frequentist statistics that you think about. Statistics as a field only goes back maybe 100 years, 1920s or so. P-values, the things that are often used to make decisions in statistics, go back into the 1930s or so less than 100 years ago. Bayes actually goes back to the Reverend Thomas Bayes, a obscure English clergyman who was an amateur mathematician and wrote down a formula for how to update probabilities in light of data. And this rule, called Bayes' rule, is the basis for everything that I do. So it's a little bit unusual to specialize only in Bayes, just because up until a few years ago, it was very difficult to do Bayesian calculations. The computing was hard, and also agreeing on what the right prior distribution. Bayesians have to have a starting place. They have to start somewhere, and then the data come in. And so this prior distribution that you have for what you think is the true state of nature gets updated by the data. And that choice is also sometimes controversial, because you and I might have different priors. You and I might not agree on what the current state of the world is. So it encourages you to think about robustness and what does the data show in light of what you already know.
Right. And its impact in terms of biopharma?
So Bayes is particularly useful for combining information, disparate sources of information. That's sort of what it's all about. And so meta-analysis, where you're trying to look at the results of several different studies, pull them together into one overall viewpoint on what's going on. That's a naturally Bayesian activity, and so I do a lot of that. Clinical trials are very often best run in an adaptive fashion. And so that means incorporating information as it comes to light, making decisions, changing the rules of the trial in the middle. Bayes makes that very natural. So anytime you want to kind of incorporate information that you've got in a more general way, rather than analyzing each data set separately one at a time and having no way to really combine all that information together, Bayes is really used for that. And regulators are pretty understanding about these things, especially in rare and pediatric disease, which is, I think, what we're going to talk about a little bit later.
Fantastic. All right. So you mentioned about clinical trials. So what are some of the uses of Bayesian statistics in drug and device development and clinical trials?
One of the things that I'm being approached about a lot lately is incorporating historical data. A lot of companies want to randomize some or even all of their patients to the treatment group. There may be ethical reasons why you don't want anybody to get the control or get a placebo. And yet you still need to know about how well the patients would do were they to get standard of care or placebo. So the company may have access to a database that contains a lot of historical information on the placebo group. And they will come to me and say, well, can we use this information? Can't we just, put all of our current patients in the treatment group and have rely on these older patients for the placebo? And depending on how rare or how important the need is for a new treatment in a particular area, that may be an acceptable method. Even if it's not acceptable to have all of your patients in your placebo group come from history, you still could cut down the number of patients you randomize. Nobody said you have to randomize 50-50 to the two arms in a trial. So you could randomize 2-to-1, 3-to-1, 4-to-1, only having a few placebo patients in your trial, and then rely on these historical placebo patients to kind of make up the difference.
Right. Okay. So you mentioned regulators a bit earlier, but how do regulators feel about Bayesian approaches?
I think Bayesian approaches weren't in common use in statistics until the 1990s, maybe around 2000. And the reason is that the computing that is necessary to do a Bayesian approach is more demanding than it would be in a traditional statistical setting. And so some regulators are just not that familiar with Bayes because they just weren't trained that way. They've been trained longer ago than say 2000. So in large, I wish I call it, in diseases where large sample sizes are still possible, breast cancer, for example, you have a lot of patients that are available to study. Regulators may be less intrigued by Bayes because they think, well, can't we just do this the way we've always done it? That's served us well for 75 years. But in rare diseases, where it's very clear that you don't have a big enough sample size to generate enough statistical power to show what you want to show, I think regulators are increasingly okay with that. Another place that regulators have traditionally liked Bayesian approaches is medical devices. Medical devices is another area where, unlike drugs, medical devices tend to evolve very slowly and gradually over time, right? This year's pacemaker looks a lot like last year's pacemaker. It's just that this year the battery is a little bit smaller, and so it's easier for the doc to implant it in your pectoral muscle, something like that. But the way it keeps you alive, the pacing mechanism, it's pretty much the same as last year. So last year's data, therefore, can be reliably used as a control group for this year, right, because you don't have a dramatic change. The placebo, the standard of care hasn't evolved so much in the last couple of years that you don't think that those older data are still relevant.
Right. Okay. So how does Bayes help with borrowing from auxiliary information? So say, for example, natural history data.
Well, so that's another, thank you for bringing that up. That's another subject that we've been working on lately, especially in rare disease, where you have patients that may be ALS patients that are progressing in their disease over time. Their motor function is slowly declining over time. You would like to make some sort of intervention and then try to show that there's a change in their rate of decline, that their decline is at least slowed down, maybe even stopped entirely, and the patients stay flat for a little while. And so you're essentially running a Bayesian algorithm where the historical your data you're borrowing now are from the patient's own natural history. And so we will fit the model based on the patient's natural history, project where they should be, were they just to continue on their current trajectory. And then once they're receiving the intervention, check to see if those new observations are consistent with their historical trajectory. If not, we know the patient was a responder. The patient responded to treatment because the patient is doing better. We can show probabilistically, this outcome we're seeing from the patient is so good, it must be due to the drug. It can't have happened just by random chance.
Right. Okay. And what is the capability of real world evidence to contribute meaningfully to the drug discovery process in Europe, US, and elsewhere?
Yeah, that's a great question. Patient's natural history data is a form of real world evidence. The patient was not randomized to anything at the time you just started watching the patient. You could imagine doing this more generally, looking at registry data, patients that are simply agreeing to be followed, but we didn't randomize them to treatment, so they're not in a trial. There are statistical methods, though, that enable you to adjust for those the lack of randomization. There's a method called propensity score matching that's very, very commonly used. Those are more dangerous methods to use because, again, you would rather have randomized data. You would rather have a way to control all those unmeasured confounders that might be lurking in your data that you can't really see. Randomization gives you an insurance policy against that.
Right.
If you don't have that, there are these statistical methods that can be used. And again, regulators are increasingly willing to accept analyses of this sort, especially if it's maybe a product that's already been approved, but we want to change the indication. We want to change the dosing of the drug. We want to change who the drug might be given to. If it's a relatively small change in the drug's approval status, regulators are willing to let us use real world evidence more and more. In fact, in the United States, there's something called the 21st Century Cures Act that you've probably talked in your blog about before, that basically encourages statisticians to be more innovative, to use Bayesian methods, to use real world data, to use more modern methods of analysis in order to take advantage of these existing large databases that are out there.
Right. Okay. And what about how do real world evidence-based results compare with those from randomized clinical trials? And in terms of regulators, how do they view those sort of results?
I think that the results can be comparable. They can still provide causal inference and causal recommendations about which treatment is better. But because they rely on existing databases and statistical corrections, I think they're always going to be not the gold standard, right? The clinical trials are always going to be the gold standard for whether a treatment is safe and effective. And so, it's a trade-off, right? We want to expose as few patients to the rigors of a clinical trial program as possible. It's a lot of work. It's an enormous amount of expense. It takes a long time. I think that it's a great idea to take advantage of real world data where we can. But I do think that for very large questions and very new indications, clinical trials are always going to be with us.
Right. Okay. So I know that's all we've got time for. So where can people get more information, Brad?
I'm going to be giving a webinar with my colleague Arnaud Monsieur at PharmaLex a week from today on Wednesday. I think it's May the 12th. And I would love for people to come sign up for that webinar and a little bit more. I think you're going to be able to help us maybe even post that link out there so people can click it and join us.
And I'll put the link above the video, of course. And if they can't make the webinar, where else can they get more information?
I'm on LinkedIn, and I'd love to talk to anybody that wants to talk about Bayes and how it can help them do a better job, especially in rare and pediatric disease, but really more generally, anybody that has data sets that need to be exploited and combined with existing data, would love to talk to you about that sometime.
Fantastic. All right, well, Brad, well, look, thank you very much for sharing that information with me. As you said, the link for both, well, your LinkedIn profile will be on there anyway, but the link to the webinar will be up there, and also to the PharmaLex webpage. If there's any information on there, that will be above there as well. So all that's going to say is, Brad, thank you very much. Have a wonderful week. I hope the sun shines in the Midwest all week today, all week from today, and you have a fantastic week. So thank you very much for joining me today.
Thanks, Rizwan. I hope the sun comes out where you are too.
I certainly hope so, although the forecast is rain for this evening and for tomorrow, I think. So there you go. We're into May now, scarily, and yet the weather is so variable. Who says climate change doesn't happen, eh? So thank you very much. And so thank you very much for you for watching. I hope the sun is shining where you are today, and it is going to shine for the rest of the week. Have a wonderful week. I hope you found this useful. As I said, check out the links. Make sure you register for Brad's webinar if you're interested, because it is an interesting topic. And, you know, as I said, there's lots of ways that people can use the data sets and utilize them. So check out the webinar and check out the links above. And until next time, as always, stay well and stay safe. Bye-bye.
Thanks, Rizwan.